There we go. All right. This talk is on recent advances in greenhouse and nursery IPM. And I say recent advances, I reuse this title, recycle this title almost annually. And because it's really based on some of the more recent research that we've done in terms of insecticide trials, maybe new findings or things that have been relevant to some of the challenges that growers have faced, or it might be also some new consolidation of some new data that again might be kind of relevant for y'all. For those of you that have a smartphone kind of handy as well, there's going to be some like live polling, some opportunity for you to kind of chime in during the presentation. If you go to pollev.com, and it's going to ask you for a little code, you're going to put in Erfan V202, and you're going to be able to contribute to a little in-presentation stuff. You'll see when you first get there, however, there'll be this little like registration for credit thing. I don't know what that is. We don't need it. So just hit that skip for now. You do not need to register, but it'll ask you just for like a little nickname. So you can just throw in a little nickname after that. And anytime a question pops up in this presentation, you should be able to answer live on your phones. So naturally, all of you are very familiar at this point that when we're talking about horticultural pest management, greenhouse and nursery pest management, we're talking about battles with mini aliens, right? So these are two-stripe walking sticks that we have in Texas. And right here on the back of this large female, so this is a female right here, and this is a male right here, is actually mating with the female. So you can see right here where the action is going on. We often see this and people think that maybe this is an offspring, but it's actually a mated couple, and the males are much smaller in this particular species. It's not uncommon as well. People will see little particles of dirt or dust kind of crawling around, wondering if they're losing their minds. And although that is possible, it's also very possible that you're just looking at a lacewing larva. You can see right here, this is a type of predatory insect. So it does have these mandibles right here that it uses to grab onto other soft-bodied insects and eat them. And they will put the carcasses of other insects on their backs as a kind of a protection and camouflage. And, it's funny that these have been making the news a lot this year, although I think we noticed them a lot more in Texas last year. And this is the flannel moth or puss moth caterpillar. And they look like little mustaches or crawling wigs. They look very fluffy. And as a result, they can be very tempting to want to hold and pet, but they are highly toxic to touch. They'll give you a horrible skin rash. So it is incredibly ill-advised to actually touch these. So something to be aware of there. So let's give this a quick little whirl here. Let's test out how this is working. So what were some of your most problematic pests this year? You're going to use your smartphone. Again, going to pollev.com/AirfunV. What is that? 202. Up at the very top of the screen. There we go. Starting to get some responses. All right. Mealybugs, thrips. And you can put more than one answer in there, I believe. So you can, if it was a major issue, you can put in multiple. Aphids, we've got fungus gnats, aphids again. So aphids definitely a very common one. Wait a few more moments. White flies, yeah, okay. These are some of our common culprits, the same things we see every single year. Spider mites, yeah. Give it another moment. This is, I think, just a nice exercise just to get y'all practicing using this here. We'll wait for maybe one more response in there. Maybe, or maybe not. All right. So aphids, yeah, absolutely. Aphids are an incredibly common one. We're going to be talking about a good number of these. So aphids, thrips, white flies, and mealybugs. Unfortunately, we can't always cover all of them, right? So I'm covering ones that I've done research in most recently, and that there's more recent data and also that can fit within the hour. Now, first starting off, right, is monitoring strategies. Okay, I hope that a lot of you are already using sticky cards or sticky traps. It can be a little difficult or tricky. Take a little while to learn how to identify things on the sticky traps. Or, for example, this is a whitefly, but actually a banded wing whitefly. So when you take a closer look at some of these whiteflies that, although can fly around, are not considered a major horticultural pest in Texas. We also have sticky tape, right? So this is kind of the same as a sticky card, the same idea, except not only is it potentially handy as a monitoring tool, but you can just see how many insects are on there. And if those are half of them females, that's that many less eggs are being laid. We also had Dr. JC spoke a couple days ago. He was talking about this beading technique. And this is a common practice, especially for things like thrips, where they might be in and or on the flowers and hard to see. In this case, I have a piece of white foam board that I like to use inside of like a plastic sheet. And you can basically hit that plant or tap it and try and get those thrips to fall out on that white foam board, and they can be a lot easier to see. From there, you can collect them in, say, an aspirator. So you're sucking here, creating suction in this vial. Very important that there is a filter in here, otherwise you're going to be eating insects. And this is where the insect goes in. So you can collect it in this vial. And that way you can see them a lot easier under a microscope. You can stick them in a freezer. So next time you want to look at them, they're not moving around, or you can send them off to someone for identification. And then never underestimate the importance of plant inspection. And this was mentioned a few times during this webinar series when people were saying you better have some kind of a hand lens. I think some of our speakers even threatened that if you do not, that you'll get a smack in the head. And so a hand lens can be pretty handy, and they can be as cheap as say about $10 for something that's going to give you reasonable magnification and some clarity. But you know what? I have not been carrying these around on my person nearly as frequently as some of these other solutions I'm going to show here in a moment. When I'm doing dedicated scouting or monitoring, I'm usually using a head lens. And the reason for this is, I mean, it's not that much of an investment, right? About $20 for one of these head lenses off Amazon. Both of your hands are free, so that way you can manipulate your plant material and look under those under the plants a lot easier than if one hand is occupied holding the lens. In the case of, if I'm just visiting a grower or just something that I typically have on my person, is either a clip-on lens. So this is actually one that I've specifically acquired myself and seems to work pretty well. This one's $45. And it has actually, so this is a macro lens right here, 15 times macro. And on top of it, which can screw on or screw off, is a wide angle lens. So you can remove that wide angle lens and get just the macro, or you can get a wider field by putting on that wide angle lens. And this just clips on your phone. So it's kind of easy to put on and take off and get some very quick magnification with your camera on your phone. And then this is actually my current preferred solution, but it's probably the most, I mean, it is the most expensive because you need to buy a special case. So you can see here right now, this is actually the case that I'm using on my phone, and it has a special slot to actually be able to slip in and twist in their lenses. And so it makes their lenses very compact, very small. You can see it right here, and very easy to basically step in and take out. And the photo quality is also really great. So this is an example of a picture I took with that particular lens on my phone being a Pixel 5, a Google Pixel 5. The only tricky part with this is that they don't make these cases for every type of phone. It's got to be one of the common mainstream, either an iPhone, a Google Pixel, some of the Samsung phones, so on and so forth. And you can see that it gives you enough magnification that you can even see whitefly eggs. So that's probably going to be sufficient for what you want in the field. All right, so now going on to a little bit more specifics, we're going to talk about thrips. So there are many different species of thrips, right? There's over 6,000 described species, and there's two different main types. One's tubulifera, which lays eggs outside of host material, and terebranchia, which lays eggs inside the plant tissue. And they are called thrips, or in this group Thysanoptera, because they have these fringed wings. So basically, these hairy wings is where they get that name from. And they will feed on immature, mature, and senescing leaves and flowers. And some of them can be predators. And this might come as a surprise. Frankliniella, Frankliniella occidentalis, which is the western flower thrips, is actually a predator of spider mites. So if you're very vigorous with your western flower thrips management program, and you notice, big spikes in your spider mite populations, that may be the reason. So there's one of two solutions: either one, only manage your thrips when you really need to, or two, if you're managing your thrips, make sure you're ready to start managing spider mites as well, if that is a common kind of co-occurrence that you see kind of happens there. In terms of their general life cycle, right, they're laying their eggs on or in the plant tissue, and then you're getting these two instars that already start feeding. They're very hard to see in this first instar stage, pretty hard to see in the second larval instar stage as well. Then they'll prepupate, pupate, and then become adults. Only the adult stages are winged, and that's when they can actually, fly around and distribute. And Western flower thrips are probably some of the most well-studied thrips. 1/3 of the total academic publications on thrips are in the last 30 years are on Western flower thrips. Now, a common question that gets asked, right, is if we have thrips and want to monitor for them, should we use the blue sticky cards or the yellow sticky cards? And, I think there was maybe at some point some preliminary information or data that suggested that blue might be more effective. But what I've seen or haven't seen, I don't know. I shouldn't give the answer away yet, because this is where you chime in. So, again, grab your smartphone. You should still be in the Poll Everywhere site there. So, again, that's pollev.com/Erfan V202. And you should be able to answer one of these. So, what color trap is most effective for thrips? A, is it orange? Or B, blue? C, yellow? Or D, some kind of a striped pattern? We'll wait for like just one more moment here, maybe for a couple more votes. Somehow we have a vote percentage that's above 100%. We have 101% here. So if someone's voting in fractions. All right. So it looks like most people are thinking blue sticky cards. And I think that has been not an uncommon perception that blue is going to be more effective for trapping thrips. However, there's not a whole lot of strong data that shows that blue is consistently more effective at trapping yellow. And this study here alone by Sarah Jandrkic in Canada, she was looking at yellow sticky tape, blue sticky tape, or a patterned blue sticky tape. And this is the average number of thrips caught every 2 weeks per 2 feet of tape. And as you can see, she consistently found quite a bit more thrips on the yellow sticky card than either the blue or the striped blue. Sticky tape. And so this suggests, that especially in this case, that yellow was actually more attractive. It's also been suggested that different shades of blue might have different levels of attractiveness to thrips. And every manufacturer provides a slightly different shade of blue. So if you want to increase the efficacy of your trapping, one thing you may consider, and you want to try blue sticky traps, you may consider buying some blue ones, putting them next to your yellow ones, and just seeing whether the blue ones actually capture more thrips either earlier or more consistently higher than the yellow sticky traps. If they do, then buy those blue ones and stick to those blue ones from that particular company. Don't switch around. If they don't, you know what? Why complicate things? Why have blue sticky cards and yellow sticky cards when you can just trap them all on one? There is an excellent field identification guide for thrips, which I have right on yonder, but you can also access the free PDF by Googling literally the title of this PDF right here. So, Pest Thrips of the United States Field Identification Guide. You'll find that free PDF. And it's helpful to know what are some of the most um, important and how to identify, I should say, the most common thrips that we see and how to identify them. So some of them, echinothrips, which is actually probably the most common one that we get on poinsettias, that at least I have seen. As the immatures, they are quite very similar color to the underside of the poinsettias. It can be very hard to see. Almost looks like a thick trichome, right? These like little hairs of the poinsettias. And it's not until they're adults that they are really dark and almost completely black and a lot easier to identify at that point. We also have Western flower thrips, which even as adults are this kind of this more beigeish type color and do have this darker part in the abdomen down here. And this is probably one of the more common ones we see on all of our other ornamentals. Now, if you're a little further down, and this is as Western flower thrips, the name suggests this is where we usually get the type of rasping damage that we see on flower petals. Now, if you go a little further down south, like Houston and further south than that, you're going to start seeing some chili thrips. And they can be distinguished by these three little eyes here, ocelli. These are three little, visual organs right here, these little three dots. And they're a lot smaller, even as adults, than all those other thrips as well. And they're beige, and the dark banding kind of goes across the abdomen like that. Now, if you have chili thrips, say, on roses, they can cause rose rosette like symptoms without it actually being rose rosette. So that means that by treating for your chili thrips, you can actually cure those symptoms of your plant. On the other hand, if it is actually rose rosette, then treating for the chili thrips will not cure it of those symptoms, right? So there's a bit of a difference there. But chili thrips can cause some kind of witch's broom type effect as a result of their feeding. In terms of preventing thrips, one of the things we want to do is cull or remove. We saw with Dr. Ann Chase, he spoke a lot about preventative plant pathogen management. A lot of the same or similar principles apply when it comes to insect management as well. So if you have very highly infested materials, you certainly want to actually bag it and get it out of there. If you're having senescing flowers on infested materials that are falling to the ground, that can be a source of infestation, right? So these thrips now are pretty excited because now they don't want to be on this declining flower and are going to look for some new material. So you're basically unintentionally concentrating some of your new flowers. So it might not hurt to prune or at least collect some of those flowers on the ground and again trash them and get rid of them. And then there's also netting, right? And especially with us down in the south, airflow is commonly an issue when it comes to insect screening, and that's why you need to build out this structure. So in this case, the structure has been built out to increase the surface area of this netting, so as to allow for sufficient airflow through those swamp coolers. But you can get thrips-proof netting, so that's netting that's fine enough that does not allow thrips to get through. And outdoors can be a major source of thrips, that movement inside. In terms of biological control, I'm not going to go into too much detail because we have a whole talk from from Miss Suzanne Wainwright, whose presentation was recorded and is on the Six-Legged Aggie website. And she spoke about biological control. But there are several natural enemies for thrips that are available. And we know that Amblysia swirsky is pretty well adapted to warmer climates. And we have tested it with whiteflies that we'll see here shortly and has shown quite a bit of promise. Now, when we're talking about the landscape, it's not really worth it to release any of these natural enemies in the landscape. At least there's no data so far demonstrating that it's really, economically feasible or that it's very efficacious, but it's more about not killing them. So it's about being cautious about what you spray. So when you're spraying very broad spectrum insecticides, if there's a lot of insects listed on, on the potential targets of a pesticide, that suggests that it's broad spectrum and it's going to hurt a lot of things, including some of these predators as well. So when it comes to insecticidal control of thrips, if there are conditions that are favorable to thrips, they can be very hard to control even with good insecticides. Some of those so-called favorable conditions includes a very thick canopy. I mean, if you have a thick canopy and you're not going to get good insecticide penetration, it's going to be very difficult to control those thrips. So good coverage is key. Want to make sure we're breaking the life cycle. So a lot of these products for thrips usually suggest some kind of a 7 or a 14-day spray interval. Make sure that you follow that, because if it's not going to work against, say, pupa or eggs, then what you're spraying, you might be killing all the adults and matures, but that next week you might get a whole new suite of insects that or thrips that need to be managed. So you want to make sure you're spraying on a interval that's going to help break that life cycle. And lastly, we want to rotate modes of action to avoid insecticide resistance. And we'll see these mode of action groups here as I show some of the pesticides. Now, again, when I talk about recent advances in IPM, I'm drawing on a number of data sources that are not necessarily my own, but are open access. So one is the IR4 project. This is federally funded, and they basically have researchers all around the country, including myself, that contribute to this database in which we're trying to help bring pesticides to specialty crops, including the horticultural industry. And there's also another publication called Arthropod Management Tests, which is short insecticide trials, replicated insecticide trials. And that's also where I publish a lot of my data as well, so that other extension personnel or even growers can access that as well. So when you're talking about thrips, I want to make this very clear. First off, mention of trade name is not an endorsement. All right. I know it's probably just a lot easier for y'all to look at the trade names. They're a lot easier to pronounce. And oftentimes there is no generic. So there might be only one trade name for it. But I've also included the active ingredient. So if it is generic, you can look for that in the market. You can also see the re-entry interval for all these pesticides and the mode of action number. So these are the mode of actions that we want to rotate when we're using different insecticides. Now, I'm going to be going through some of these tables here. You're more than welcome to take screenshots of this. You're also more than welcome to go back to the recording and take screenshots of it. So this is, y'all are welcome to copy this as you wish. But at this moment, I might not be going through it slow enough for you to write it all down. So I just want to give you that heads up. But just from looking at this, you can see a few insecticides that show some promise. So Ancora has one study that showed good control against thrips. We got here Avid that has good to excellent control of thrips, and that's more than 10 separate studies. So that's pretty consistent, showing that it works quite well. We also got Botanigard or Bioceres or Mycontrol. These are all Bovaria bassiana products to give good to excellent control, and that's 6 studies. And we also have Conserve or Entrust. And the active is Spinosad, which gives good to excellent control. And again, more than 10 studies. Want to be very careful with products such as Spinosad, any of these actually, but Spinosad particularly, because this is one of those that was a little overused in the country just north of us. And so there's a lot of thrips populations that are resistant to Spinosad. And so they lost this tool that is good to excellent. And so again, you want to make sure you're not spraying this over and over and over and over again, maybe 2, maybe 3 times consecutively. But after that, you really want to rotate to a different mode of action group number. Some others here, Hachi Hachi is consistently good to excellent. We also have Cyantraniliprole or Mainspring is also consistently good to excellent. Also have Overture, which provides good to excellent control. And lastly, Pradia, which is a relatively new product from OHP that so far, only 2 studies, but so far has demonstrated very good promise against thrips. And in our last group here, Pylon. Pylon is pretty consistently considered like an industry standards, and that has good to excellent control there. And you can see some others here, for example, such as Expire, that provides mediocre to excellent control in 6 studies. So there may have been 1 or 2 studies in which it did not work that particularly well. We have that same kind of situation with Sarisa, where we have more than 10 studies and it's mediocre to excellent. So really, sometimes it might depend maybe on the Thrips species you're dealing with, which again might help with identification. Might be related to penetration. How thick is that canopy, so on and so forth. Now, if you want to know how to look up these studies yourself, I have made a number of tutorials on YouTube for how to interpret that IR4 database for insecticide efficacy. This is a 2-part series that you're more than welcome to watch. Now, going on to mealybugs. Mealybugs are in this family Pseudocosidae, which Cosidae is soft scale. So Pseudocosidae is like fake soft scales. So what makes them fake soft scales? Well, soft scales, the females only lay one clutch of eggs. They basically die in their little ovisac when they lay those eggs. Whereas mealybugs, the females can lay multiple clutches or multiple groups of eggs. And so y'all are probably quite familiar with what mealybug eggs look like at this point. So that is kind of like an ovisac of eggs right there. Here are some immature mealybugs. So they don't have that white waxy surrounding them quite yet, and they are relatively quite considered most mobile kind of in that first stage, although they can be mobile thereafter as well. Then we have a number of different species, such as the long-tailed mealybug, aptly named because of the long tails. We have citrus mealybugs and pink hibiscus mealybugs. You can see here's another photo of those pink hibiscus mealybugs as well. And this is citrus mealybugs actually on hibiscus, so not incredibly common, but we did find them able to infest the buds and flowers, specifically of hibiscus. They were not infesting any other parts of the citrus. All right, so now new number, who dis? So just based on those images we've seen, who do you all think this is? So use your phones again to answer. Do you think this is the Madeira mealybug, the citrus mealybug, pink hibiscus mealybug, or none of the above? All right, so we got pink hibiscus mealybug is currently in the lead, followed by Madeira mealybug and none of the above. All right. So whoever said none of the above, um, which, as I guessed, was Suzanne. I can see in the chat. Is It was none of the above. That is correct. This is actually not a mealybug. This is the immature stage of a lady beetle. Now there are some known as mealybug destroyers. Here's an example of one. This is the adult mealybug destroyer, and it's eating a mealybug. It is actually not eating its own offspring. It looks very similar to a mealybug, and it's thought that they look like mealybugs as it matures to be kind of like a wolf in sheep's clothing. It's a way of being able to get close to their prey without their defenders. So ants will actually help defend and herd mealybugs because they like their honeydew, that sugary solution. And so by looking like them, they again are like a wolf in sheep's clothing. They can get close to their prey and then eat them. So one way of knowing the difference is you don't get really colonies of these mealybug destroyers. You don't get colonies of these predators. And also they're hunters. They move around quite a bit looking for prey. They're rarely just sitting there. They obviously don't produce honeydew because they're not sucking on the plant juices. And you will eventually start to get some of these adults, which are winged as well. Now, very important, and this has come up a number of times, is mealybug sanitation. So if you've had highly infested plant materials with mealybugs, and so you go to get rid of that plant material, but you're just getting rid of the plant material and maintaining the pots, be aware that those mealybugs can stay on there and become a potential source of infestation next time you put plants in there. Now, we don't know, I have not been able to find data on how long it is that they can stay on there without plant material and still potentially be viable. This is what we would call potentially diapause. So it might be that when there are no sources of plant material nearby, they can essentially go in a state of metabolic rest. So they are basically asleep, like they're hibernating, waiting for some plant material to come there, and then they can reactivate. And so we do have some infested pots right now that we have sitting in a cage. In early spring, we're going to put some plant material in there just to get an idea of whether, it is actually possible for them to survive, say, an entire cool season, without any food, and then really become activated again and feed on plant material. But we do know that this is a common issue. So if you are reusing pots, and especially they've been infested by mealybugs, make sure you are either A, sanitizing those pots, or B, strongly reconsider reusing pots. Now, we did do a trial with citrus mealybug on hibiscus. And so here are some of the insecticides that we use. So one treatment here is our untreated control. So we, sprayed nothing. But then we had some different formulations. These are all products from BASF. So Ventigra, Velifer, which is their Bovaria bassiana, and combinations of ultra pure oil and Ventigra, or ultra pure oil, Ventigra, and Velifer. And we're using Safari as kind of our gold standard. Our dinotefuran is known for working very effectively against mealybugs. And you can see here an idea of application dates. So most of them were applied multiple times. So you can see, for example, Ventigra was applied twice on a 2-week interval. And again, you can go back and look at this video or take a screenshot if you wish to record these details. So I want you to pay special attention. So you'll notice here on the x-axis, I have days after treatment, starting with day 0. So that's when the first treatment applications were applied. And here on the y-axis, I have mean immature mealybugs per plant. So you'll notice here right off the bat that all of them are starting with relatively similar numbers of immature mealybugs. There's some ups and downs, but when you look at the variation around that mean, they're all overlapping quite a bit. 7 days after, we don't see too many things teasing apart, and there's still a lot of variation. So I'm going to draw your attention more to days 14 and 28. And the reason why not 42, well, if you look at the untreated control, it also decreases drastically by day 42. So it's really hard to make any treatment comparisons on that particular day. So let's just exclude that when we're actually looking at this graph. So if we look here at day 28, you'll notice that our Safari worked incredibly well, right? So you have very low mealybugs as compared to the untreated control. We also see now Ventigra alone provided some reasonable suppression, but we got some very good control when we're looking at ultra pure oil with Ventigra or ultra pure oil with Ventigra and Velifer rotation. So in other words, the treatments that had ultra pure oil in that rotation worked very effectively. If we look at now the mature mealybugs, so there's the same treatments, but now looking at matures, again, we can see, pardon me. This is what I get for trying to get all fancy here with annotations. All right, so if we look at the matures, you can see, again, Safari working very well. We can see our ultra pure oil, especially right here. Look at that. That's the ultra pure oil, Ventigra, then Velifer. That provided very good control of our mature of our mealybugs. So we didn't see any matures basically by day 28 after the treatment application. Now we have another set of treatments, and this is all products from OHP, specifically Pradia. All right, so this is just different. You'll notice different treatment rates, and also either as a drench or as a foliar application. And so let's focus on that day 28 again for the same reason as before, right? So our untreated control is nice and high. So we have a lot of good comparisons to make here. So we can see again, our Safari worked quite well. Obviously, it's the same Safari as that last graph we saw. Now you can see here a Pradia either applied in a medium or high rate also provided very good suppression, very good control of the mealybugs. And the drench also provided very reasonable suppression as well. So actually the variation here overlaps with the Safari. So it's arguably quite similar, although perhaps not quite as good as a Safari. All right, we see the same thing now. If we look at the mature mealybugs, we can see again Safari working quite well. We can see here the Protea drench that's kind of overlapping a little bit with the variation we're getting in the Safari, and we're getting some suppression from the foliar applications. Now, in this case, it's not quite as good as, say, a Safari drench. So it might be something that would be used in conjunction with say a Safari drench to provide some additional control if that is something you were needing. So when we're looking at actually summarizing some of the mealybug data that is available out there, some of the products that we see good to excellent control with Aria, good to excellent. We see Discus provides good to excellent. Now you'll notice this group 4A, for those of you that cannot use neonicotinoids, avoid 4A. All right, so you cannot use those ones, which includes Flagship and Marathon as well, which are also 4A and provide good to excellent control. We do have Protea, again, which so far is excellent based on one publication in arthropod management tests. Rycar appears to provide some good control based on 3 different tests. Again, Dinotefuran, we already know, provides very good control as a drench, is excellent. As a spray, can also provide good control. We also have TriStar and Ventigra, which also appear to provide excellent to good to excellent. Again, TriStar being a 4A, but Ventigra is not. So again, that gives you another option of something that can work quite well against mealybugs. We have a few other options. Safer insecticidal killing soap. All right. Excellent control as well. Now, with something like this, I would caution that it's very important to get good coverage because that's something that's going to have to actually contact them. All right. And then some of these others are considered mediocre to good for mealybugs. Again, just based on the number of studies. So 2 studies, 1 and 2 studies, so on and so forth. Some of these others are considered not maybe the best for mealybug control based on currently available data. Now going on to whiteflies. That was another of the very common pests that everyone kind of listed. There's over 1,300 species of whiteflies described. The one that I have unequivocally seen the most common here in Texas is the sweet potato or silver leaf whitefly. And it's referred to as a Bemisia species complex at this point. It's not a single species. There's actually multiple physically or morphologically indistinguishable species. They all look identical, but have some very different characteristics. So we used to have this A biotype originally, and then we had this invasion of the B biotype, which could replicate a lot faster, had much broader host range. And as a result, that's when whiteflies really started becoming an actual problem for us. And then more recently, we got this Q biotype, which is now referred to as MED, and B biotype is referred to as MEM1. And this biotype is marked by a lot of insecticide resistance. So when we look at these 3, so New World, this is the original that we had, you can see the host plant range was very narrow as compared to this MEM1, which was also known as our B biotype. So this was our B and this was our Q right here. And then you can see the biotic potential is how quickly they reproduce. So you can see also, again, this B reproduces much quicker than that Q. And then in terms of carrying plant viruses, it was also more, and also a higher propensity for insecticide resistance as compared to the A. Along comes Q, similar host plant range, much lower biotic potential. So they actually don't reproduce quite as quickly as the MEM1. And what does that actually mean in a practical terms? Well, if you don't spray insecticides, this B biotype will actually outcompete the Q. And so you'll end up with a population that's a lot more susceptible to insecticides. Now, of course, you don't want to spray nothing because that's going to be also problematic. But it's very important for this reason that you spray judiciously. You spray when you need to, so that you are not inadvertently promoting a population that is resistant. So you'll notice here at the very bottom, in terms of resistance, this Q biotype has some major insecticide resistance. Specifically, all right, and this is an excellent publication from the University of Florida. We're looking at, again, MED, which is the Q or the MEM1, also known as the B. You can see here that these particular insecticides, pyroproxyfen, um, pymetrozine, kynoprene, and buprofizine, that work quite well against the B biotype, do nothing against the Q. So again, you have to be very cautious. They do so little that actually, when there's research labs trying to maintain a colony of whiteflies that's only meme one, they will spray pyroproxyfen over and over and over again, because it'll decimate this Q biotype and do nothing to the bee. I'm sorry, the other way around. It'll decimate the B biotype, but do nothing to the Q. So in that way, they can maintain their Q type population. In terms of their general life cycle, whiteflies are laying eggs, 70 to 80 eggs per female. It takes, anywhere between 5 to 22 days, depending on temperature, for that to emerge this first instar nymph, which then becomes a second instar and a third instar. And then they undergo this fourth instar, which also has this pupal stage in it, which then it stops feeding. You'll see these little red eyes, and out comes a new adult. So, one of the things that we noticed when we were scouting for whiteflies on poinsettias. So we did a commercial trial, where we looked at 6 different greenhouses at 3 different locations, and for the full poinsettia growing season. And we would look every time we visited those greenhouses, we would inspect 50 different poinsettias. So for a total of about 3,732 poinsettias. Of all of those poinsettias were inspected, you'll see more than, it's about 3,300 or 3,400 of them had less than 5 whiteflies. So for the most part, even when you have an infestation, usually it's very highly kind of concentrated, right? It's not homogeneous. It's not even throughout an entire greenhouse. You'll notice here some like tiny little blips at the 70 and 75. All right. So usually you get very high populations in pockets. And that's why it's so important that when you're scouting, that you're trying to randomly sample throughout each house, so that you'll hopefully catch some of these pockets. Because if you just look in one corner, there's a very good chance you might just be seeing some of the very low populations and not catching where those hot spots are. And we've already spoken about insect screens, something to consider when it comes to whitefly management as well. There is a number of biological control agents available for whiteflies, 2 of which we tested this last year. When I say this last year, I guess it was actually in 2019. And this predatory mite, Amblysia swarskyii, and Eretmossus rhymicus, a parasitic wasp, which have promised to work together to suppress whiteflies and also work at higher temperatures. Additionally, this Amblysia swarskyii can suppress thrips as well. So if you have a Thrips, they can feed on their eggs and first instars to provide some suppression of those as well. So we did this trial. These parasitic wasps are released as these little pupal cards, as you can see right here. And you can see these are each individual little pupae, and wasps are going to emerge and then start looking for whiteflies. Whereas the predatory mites were released in this carrier material. You can see right there. So it's a little fan that's basically blowing that carrier material with the mites onto the plants. So pretty quick, relatively simple distribution mechanism. You don't have to worry about PPE. You don't have to worry about REI. You don't have to worry about phytotoxicity. And those were some of the benefits that we were looking at as a potential incentive for considering biological control. So you can see this is what it looks like when you have a lot of that carrier material on there. It typically wouldn't be that much. And usually, you'd have practically none on there by the time it's going to market. So it would not be very noticeable. Now, what did we see in terms of the data? So I'd mentioned that there were 3 growers, right? So we have grower A, B, and C. On the x-axis, we have the week number of the growing year. And on the y-axis, number of immature whiteflies per plant. Now, based on some data collecting we did at some retailers, we found that there's this threshold of about 75 nymphs per plant. All right. Above that was very uncommon at the retailer. But anything below that, I mean, even at a florist that was selling poinsettias for $50 a pop, had up to about 75 nymphs per poinsettia. So we were considering that to be our retailer threshold, that we need to maintain levels below that. You'll notice that for all of these, by the end of the trial, all right, we have numbers that are below that. All these dots and lines are all below that threshold. Now you'll notice, so IPM is our biological control, that's the orange, and the blue is the insecticide. So we also had another greenhouse where they were just using conventional insecticides. You will notice the IPM house tended to have some plants that typically had higher numbers than, say, the chemical house, but eventually they do end up down low, kind of close to each other. And this is doing releases of natural enemies at a rate that's economically comparable to insecticide inputs. And again, that's not considering perhaps the economic cost of having to wear PPE or having to train employees or REI or loss due to phytotoxicity. If we look at the proportion of poinsettias infested, again, based on some of our retailer data, if we ended up with less than 70% at the end of plants having any whiteflies, then we were kind of good to go. You'll notice that throughout our entire trial, the number of poinsettias that had 70, sorry, we never found more than 70% of the plants to have whiteflies in a single visit. So of the 50 plants that we scouted, never did we find 70% of them to have whiteflies. You can see here we get pretty close, but not quite. But you will notice this trend that on average, the IPM or biological control house does have higher numbers of whiteflies than the conventional house. But this is kind of our first run. So it is possible that with further optimization, you can kind of get them to be a little bit tighter, a little bit closer. You'll notice that by the end of the trial, they are typically pretty similar in their proportion of plants infested with whiteflies. Now, in terms of insecticidal control for whiteflies, again, good coverage is vital. We want to rotate the modes of action, break the life cycle. So again, we do have pupae and eggs here. So we want to make sure that if we're spraying and that spray is specific to the life stages that are feeding, we might have to spray again because those pupae and or eggs are emerging and be feeding there shortly. And also be aware of this meme1 and med biotypes or cryptic species, because you want to avoid that insecticide resistance issue. Now, we did do a couple trials this season as well. So one was considered a cleanup trial. So imagine you just grew a crop that had whiteflies. You've now emptied that greenhouse. You're about to come with a new crop. You don't want those lingering whiteflies to go on this brand new crop. So what we did was we sprayed. We first introduced adult whiteflies into a cage, and then we would spray that cage either with nothing, with our fantastic Overton water, or with 2 fluid ounces per gallon of Clean Grow. And then after that spray application, we introduced a poinsettia. And then we counted at 3 days and 14 days after the introduction to see how many whiteflies we ended up with on those poinsettias. And you'll notice that if we did nothing or sprayed water, that already by day 3, we're starting to get about 3 nymphs per plant. Whereas when we apply that Clean Grow, we essentially end up with very clean plants. So by spraying that Clean Grow, it is possible that you can kind of clean up or prevent pest problems. Now, this might look a little bit different, right? So we're working in a small cage. It's very easy to do a broadcast application in there. This might look a bit different in a large greenhouse setting, right? So the question is, do you need to fumigate that whole greenhouse or just spraying around the benches? That yet we do not know. All right. Now, in terms of, we also tried a quick little trial to see whether Clean Go would provide protection, or could actually be used as, if you're including it as an additive in your spray tank, can it also provide control of whiteflies and as uptake? So Clean Grow and uptake are the same product. So in this case, we have Clean Grow, but it's actually say uptake, and at 0.13 fluid ounces per gallon. And you can see it does not really provide suppression. So Clean Grow is right here. You can see that the number actually goes up even a bit higher than the untreated itself. So don't rely on uptake to provide suppression of the whiteflies. Now, again, going through some of the data that's kind of currently available, things that provide good to excellent control include Avid. We have Oberon and Pede, which again is going to be very important to have very good coverage. We have Rycar, Safari, and TriStar. These are all products that are going to work very well against both the Meme1 and the Med biotypes. Now, in this case, we're going for some that are going to be really more effective for the Meme1 than Med. And so you want to be careful with the frequency at which you're applying these because you might be promoting Med populations. This includes Flagship, Solero, Distance, Marathon, Sandmite, Azotin, and Talus. And then lastly, we have Aria, which works well against the Med, but not against the Meme1. And then we have some others that don't seem to work really well against either biotypes. We also did an aphid insecticide trial, and this is just to provide, again, just some more data. And I think this is actually a pretty helpful one where we tested a number of formulations of Protea, either as a foliar application or as a drench. And we also, again, did that clean grow, basically uptake as an additive. And in this case, the question was whether we could provide some long-term residual control of aphids, say like in hanging baskets. So if you do a single application, going to get good long-term control. So we actually did this trial 2 separate times, once in the summer and once in the fall. So here's the summer trial. You can see all the, well, let's first start off with our untreated control. Our untreated control is this pink that goes sky high. So by 14 days after our introduction, we only introduced 8 adults within each cage. We have 250 aphids. So that's how rapidly aphid populations can increase when they are not left in check. The clean grow in this case appeared to provide some reasonable suppression, but definitely not considered control. When we look at all of the Protea treatments, they provided basically very good control, practically 100%. So practically completely removes the aphids from that plant. Now, this trial gets a little bit more interesting right here. So we redid this again in the fall. So again, you'll notice the untreated control by week 28, it has about 1,700 aphids, right? Again, so starting off with 8 adults per cage. You can see the clean grow this time did not appear to provide any suppression at all. So again, don't rely on it or uptake to provide control. They appear to come down, but what's actually happening here is I need to remove these because all of the plants are dead except for 2 for each of these. And so the plant is in great decline. They're basically dead. Now, if you notice, Protea, again, very low, right? Very good control. And actually, at this specific point right here, we introduced, I think, oh yeah, I don't need to annotate all this. I introduced 8 additional adults, all right? And the idea here was to see whether, even though we're not going to respray with Protea, do we still get, no aphids in the end? And you'll notice, despite the fact that we introduced 8 more adults again, there's no aphid population growth. So Protea appears to provide, based on our data so far, very good residual control, even past 28 days after treatment application. All right, so I think I'd kind of included this slide set here just in case we had extra time, but I think we are kind of close to the end here. So I'm just going to go straight to this here. For those of you interested in learning more about kind of like some of the horticultural research, I co-host a podcast with this Vikram Baliga right here from Texas Tech called Jolly Green Scientists, where we digest a research paper every single time that's related to the horticultural industry. So you might find that interesting. If you're just interested in insects or bugs, I have this podcast called Talking Bugs, where I interview an entomologist and talk about some of their most recent research. And with that, that concludes my talk. If y'all have any